Recombinant DNA technology, commonly referred to as genetic engineering, is a powerful tool in molecular biology that enables the modification and manipulation of DNA. This process involves combining genetic material from different sources to create new DNA sequences with desired traits. The technology has revolutionized fields such as medicine, agriculture, and biotechnology. The isolation of plasmid DNA is a crucial step in recombinant DNA technology, enabling the study and manipulation of genetic material. There are six methods of the isolation of plasmid DNA: alkaline lysis method, boiling lysis method, enzymatic lysis method, silica membrane or spin column method, cesium chloride ethidium bromide gradient centrifugation, magnetic bed based isolation. This process involves the extraction of circular DNA molecules from bacterial cells, which often carry genes of interest. The procedure begins with the cultivation of bacterial cells in a nutrient-rich medium to achieve optimal growth. Once the desired cell density is reached, the cells are harvested and subjected to lysis using an alkaline solution containing sodium hydroxide and SDS. This step disrupts the cell wall and membrane, releasing cellular components including plasmid DNA. Neutralization follows, where potassium acetate is added to precipitate chromosomal DNA and proteins, leaving plasmid DNA in solution. The mixture is then centrifuged to separate the insoluble debris from the plasmid DNA containing supernatant. Further purification is achieved through ethanol precipitation or silica column based methods, ensuring the removal of residual contaminants. The isolated plasmid DNA is now ready for downstream applications such as cloning, sequencing, or gene expression studies.
I'll walk you through the fascinating process of introducing a gene of interest into a plasmid, an essential technique in molecular biology. Let's dive right in. First things first, let's get an overview. This process involves three main steps: isolating the gene of interest, cutting both the plasmid and the gene with restriction enzymes, and finally ligating them together. Sounds simple enough, right? But let's break it down. Step 1, isolating the gene of interest, dude. Imagine you're in a lab extracting DNA. This DNA could come from genomic DNA or cDNA synthesized from mRNA. To amplify the specific gene, we use a technique called polymerase chain reaction, or PCR. PCR is like making photocopies of a specific page in a book. It's precise and incredibly efficient. Now on to step 2, cutting with restriction enzymes. Think of restriction enzymes as molecular scissors. They cut DNA at specific sequences, creating either sticky ends or blunt ends. This is crucial because it allows the gene of interest and the plasmid to fit together perfectly, like puzzle pieces. Moving on to step 3, ligating the gene into the plasmid. Here, the cut plasmid and the gene of interest are mixed with an enzyme called DNA ligase. DNA ligase acts like glue, sealing the DNA fragments together to form a recombinant plasmid. This recombinant plasmid is now a powerful tool for genetic engineering. As the bacteria multiply, they also produce the protein encoded by the gene of interest. So why is this important? Introducing a gene of interest into a plasmid is a foundational technique in biotechnology.
Hey guys, quick biochemistry basics here. Let's talk about agarose gel electrophoresis. Agarose gel electrophoresis is a technique used for separation of DNA, RNA, and sometimes large proteins. It is widely used for the separation of DNA. Depending on the size of DNA which is to be separated, the concentration of agarose is kept between 0.8 to 2%. Agarose is prepared by dissolving agarose in an appropriate buffer. Usually TAE or TBE buffers are used. The same buffer is also used in the buffer tank to run the gel. The mixture of buffer and agarose is boiled for a few minutes The boiling ensures that the agarose is uniformly dissolved in the buffer. The agarose is then mixed with ETBR and poured in the tray. Once solidified, the gel can be used for electrophoresis. For analyzing DNA samples, about 2 to 5 microliters of sample is mixed with a drop of bromophenol blue and loaded in the gel. Bromophenol blue is a dye used to monitor electrophoresis. The electrophoresis is carried out at an electric field of 5 to 10 volts per centimeter. Remember, electric field is the applied voltage divided by the distance between the electrodes. The direction in which the DNA runs is from negative to positive. Once the run is complete, the presence of DNA can be visualized under UV transilluminator.